We move on to stem rust. Just a nice picture of the pustule sporulating. An interesting one, these pictures were taken from herbarium samples more than 100 years old. So although the spores are dead, they still maintain their structure and they do not desiccate, or they appear as if they were collected yesterday. And they're of course still usable for DNA isolation and comparison with current day isolates. Using just a normal stereo microscope, we can also view these quite easily. And there's just another close-up of the Iridineospore of Puccinia graminis. And there you can see a germ tube coming out of one of the germ pores, and then it's already germinated from one of the other pores in the center of the spore. This picture is from one of our PhD students, Howard Castellane, and he works on adult plants, and to see the infection process on the actual stems, where most of the histology work, histological work, has been done on seedlings. So we wanted to see what actually happens in real life and on the adult plants, and I was quite amazed to see how the germ tubes branch once they come out of the spore.
So this is the leaf sheath?
This is the leaf sheath, yeah, the last internode, yeah. So the moment the spore germinates, the germ tubes branch. And I think that must also be some kind of an ecological or evolutionary adaptation to increase the chances of one germinated spore finding a stomatal opening and to penetrate and then to perpetuate itself. And there's a close-up of that section. You can see the aprosorium already collapsed at this stage. I assume this would be a 24-hour after inoculation picture. One thing I don't know is what happens to the nuclei. Where do they go? If the spore has two nuclei and it branches, do the nuclei also divide, and which fork in the road do they take? I don't know. And even 6 hours after inoculation, you can see it's quite difficult to find these on the stem, so we don't have too many pictures, but I think it is possible even 6 hours after inoculation on an adult plant that you may get penetration and even Haustorium formation. So it doesn't take that long for the infection to happen under ideal conditions. Also 6 hours, and hopefully this guy managed, well, we killed it of course, but if we left it, it would have, I think, reached that stoma quite quickly. In this case, it's a better picture than the others because the germ tubes are still intact and not collapsed. Interesting aprosorium shape. It's not as well defined or as round or as the leaf rust one. It's more of an oval shape. And there's a better picture of what it looks like before it, while penetrating, I think. And also on adult plants, if you use fluorescence microscopy and specific stains, you can follow the process from the spore, the branching of the germ tubes until it forms the aprosorium over a stomatal opening. Now, I also just took these pictures to see what happens in an adult plant. I suppose a lot of this work has been done, but we're really interested to see how adult plant resistance differs from susceptibility and how the fungus grows. So he started to optimize the techniques to do this. So you can see this is a cross-section embedded in wax and then cut with a microtome and stained, normal stains. So you can see the rust colonies in between the vascular bundles at certain points in the stem. And if you take a close-up, you can see that it grows, the stem rust pathogen grows in the area between the veins or the vascular sheaths, and that the sclerenchyma tissue of the surrounding the vascular bundles, the xylem, And the phloem actually restricts the fungus to then grow through the vascular sheath. So I think the damage of stem rust is just caused by the reduction in photosynthetic area of the stem rather than a mechanical breakdown of the vascular system itself. The same system, just a different stain, and again you can see how the colonies show up in the cross-section of the stem. And again a close-up, you can see exactly the same thing, the vascular bundles there, vascular sheaths, and then the sporulating pustule in between. So I think we're almost there. If we now take a real-life example, you can see what we saw microscopically. You can see the veins running on the leaf sheaths, and then the stem rust fungus growing in between those. And when sporulation is profuse, it almost appears as if the entire stem is covered. I think that just depends on the size of the actual pustule and the amount of sporulation that we see. But they still maintain these longitudinal growth between the vascular sheaths. So I think that's what I have to tell you in terms of the infection process and what I know and what happens with the three different rust pathogens.